It worked earlier.
Is it plugged?
Yeah.
It's plugged.
Yeah. No.
Try again. Maybe plug and unplug.
Oh, come on.
It's okay. It worked before. We'll make it work.
Yeah, no, it's fine. Yeah, it worked.
Try another one. No, it doesn't.
Oh, no, it's doing something now.
It's
No, give it a sec.
Yeah,
there
it's
we go.
found it.
Yeah.
Found it.
It did it this time.
Found it.
There we go. Unplug it, plug it in again. Great. Oh, wait a minute. Why is it not there, though? Oh, there we go. Okay, cool. Awesome. Thank you very much, everyone. It's an absolute pleasure to be here. I was just kind of reminiscing that ICAC was one of the very first conferences that I ever went to as a graduate student back in Magdeburg, I think in the late 2000s. And I've been to every one since. It's a fantastic community. It's a fantastic conference. So it really is an absolute honor to get to talk to you guys. I'm also at a very kind of exciting point in my tenure. I've had my lab for around about 5 or 6 years now. And a lot of the projects that we started early on are really starting to come to fruition. So a lot of the stuff I'm going to be talking about today is either unpublished or we're in the process of putting together preprints or have recently submitted preprints. So please come harass me. Constructive criticism is always welcome. I'm going to talk about sensory-guided behavior, and I always like to start with this somewhat silly example. This is a fish show. This is arguably one of the greatest sensory environments in the world. Everyone thinks I'm joking. I'm really, really not. I like to show this just to make the point that sensory-guided behaviors are completely ubiquitous, right? Auditory information and sensory information in general is always associated with distinct behavioral outcomes. So in a silly example like this, if Trey Anastasio busts into an amazing guitar solo, you're going to turn your head and focus your attention on him. If they bust out a song that they've not played in 20 years, it's going to elicit an emotional reaction. And what I find really, really interesting about these kind of examples is that the auditory system is not a hub of emotional processing. It's not a hub of motor action. Arguably, it's not even a hub of decision making. So in order to try and understand how purposeful behavior like this is coded in the brain, we really need to understand how auditory information flows through brain-wide circuits. And to kind of further complicate things, this is an example of almost 1,500 neurons in the auditory cortex. Not one of these neurons is actually faithfully encoding the incoming acoustic waveform. So to really understand this kind of mess of activity, my neuroscientific world point, so to speak, is that we really need to understand the identity of the cells that we record. We need to understand their upstream and downstream connectivity. We need to understand the kind of information that distinct cell types can process in order to understand how we can perceive complex environments such as the one on the left. So that's a somewhat long-winded way of saying that a key focus of my lab is to really understand how the auditory cortex is hooked up to the rest of the brain. And this leads me on to the kind of topic of descending auditory cortical circuits, so excitatory diversity within the neocortex. So what goes up must come down. This is a kind of schematic here of the 3 main efferent projection systems that are found in the auditory cortex. This is also a kind of ubiquitous circuit motif that's found throughout the neocortex, but I'm just going to focus on the auditory system here. So you can break excitatory projection neurons down into 3 main cell types, schematized on the left. So we have IT, ET, and CT. IT are intratelencephalic neurons. These are neurons that actually reside in every cortical layer. I'm just illustrating them in layer 5 here. These are the neurons that project within the telencephalon. So these are your cortical neurons, corticostriatal neurons. They project both ipsilaterally and contralaterally in the brain. On the right-hand side, these are straight-up corticothalamic neurons that are found down in layer 6. As the name suggests, these neurons project exclusively to the thalamus, and indeed in the top right-hand side there, you can see the medial geniculate body labeled quite beautifully. I'm going to spend a lot of time today focusing on these neurons in the center. These are layer 5 extratelencephalic neurons. These are my favorite neurons in the entire brain. They provide the only direct route of information flow from the auditory cortex to subcortical stations within the brain. So these are the neurons that will contribute to the corticocollicular projection in the midbrain. They also project to the auditory thalamus. Indeed, these neurons also project all the way down to the level of the cochlear nucleus, as well as targeting areas outside of the traditional auditory system in the striatum and the amygdala. So just to give you a kind of one-slide example of some of these IT circuits, so these are the intraencephalic neurons. This is just to make the point that these IT circuits really do propagate sensory signals very, very broadly outside of the auditory system as well as within it. So this is just a representative coronal section, a little bit rostral to the primary auditory cortex, and you can see the corpus callosum lighting up to illustrate that there are some really nice contralateral projections. But these neurons are also projecting very, very heavily to the striatum and the lateral amygdala there, as well as somatosensory cortex, posterior parietal cortex, and retrosplenial cortex. But now moving on to the really cool neurons, these ET neurons, something that's really remarkable about them is that they have this dense divergent processing. So these neurons collateralize to multiple downstream targets. And we can see this using an intersectional viral strategy that I've schematized on the left there. So this is an approach where we inject a retrograde Cre recombinase into the midbrain. That will drive selective expression of Cre recombinase selectively in the corticocollicular projection, so that we then hit the auditory cortex with a Cre-dependent green fluorescent protein. You can selectively label these corticocollicular neurons and then scan the entire brain and find out where else the axons project. So as you might expect, we're selectively labeling the corticocollicular projection, and indeed the inferior colliculus, the external nuclei, and the lateral shell lights up really, really nicely. But you can also see that these neurons are collateralizing into these other targets that I've labeled on the right there. So the MGB, you can see the external parts of the MGB lighting up, and you can also see that these neurons are collateralizing into the striatum and lateral amygdala. And just a quick one-slider about some kind of future work in the lab. I like to call this approach inception with cells. We live in a really, really cool era of systems neuroscience at the level of the mouse, where there are a lot of different viral and genetic tools that we can use to be incredibly, incredibly specific about the cells that we want to target. And Cre recombinase is but one recombinase that we have access to. And you can use additional recombinase strategies. Here I'm illustrating flip recombinase, to pull out subsets of subsets of subsets of neurons. So in this particular example, we've already expressed Cre in the corticocollicular projection. You can then use a Cre-dependent retrograde flip, which is a little bit of a mouthful. But what that essentially does is pulls out a subset of those already Cre-expressing cells that project to the colliculus, in this case to identify the subset that also projects to the thalamus. So these are incredibly powerful strategies. You can be very, very specific with expressing useful things in ET neurons with very distinct organizational motifs. I just want to focus on one final little anatomical finding. This was a kind of COVID project that we engaged in. What we wanted to do was use a transsynaptic tracing technique. This is just a genetically modified rabies approach to identify the local and long-range inputs to different auditory cortical projection types. So I don't have time to go into too many of the details, but this is a pretty standardized approach in systems neuroscience where we exploit we essentially inject a combination of viruses where we can turn a selection of Cre-positive cells into starter cells on which the rabies virus can explicitly act and identify presynaptic inputs. And what's remarkable is that all three of these cell types really receive information. They integrate information from a whole array of diverse local and long-range inputs. I'll also add that this has been carried out at a far greater scale at the level of the visual cortex by the Allen Institute. So this is something that seems to be pretty ubiquitous across sensory areas as well. But as you can see in this diagram, we're kind of moving from the caudal parts of the brain to the rostral parts of the brain. You can see the thalamus is lighting up in all cases, but all three of these cell types are integrating information from other sensory areas, the level of the visual cortex, the somatosensory cortex, but they're also receiving lots of cool inputs from association areas of the brain and frontal areas of the brain. So anterior cingulate and motor cortex, as we heard from Patrick's talk, project heavily onto these neurons, as well as areas further forward at the level of the orbitofrontal cortex. So combined, these neurons essentially integrate lots and lots of inputs, and they have the ability to then broadcast the results of complex computations brain-wide. And so my lab really spends a lot of time trying to understand cell type-specific contributions to auditory-guided behavior. Although we use a lot of different tools and techniques these days, we're primarily very imaging-focused, and we've spent a long time trying to develop approaches to carry out two-photon imaging deep into the cortex, so deep layer two-photon microscopy, so that we can access these projection neurons down at the level of layer 5 and layer 6. And here I'm just highlighting our ability to do that. On the left-hand side, we can use similar intersectional strategies to those that I've described before to selectively express GCAMP in populations of neurons. And here I'm showing a nice Z-stack from an NTSR1 Cree mouse line. This is a Cree mouse line that provides us with genetic access to the layer 6 corticothalamic projection type. And here we can image down at around about 650 microns below the surface and readily elicit some nice calcium responses to sounds. Just a little bit of foreshadowing. This is also some other stuff that my lab is kind of interested in, although I'm not going to present any results on this today. Holographic optogenetics is an incredibly powerful tool. This is an approach where you can use a technique called spatial light modulation to split a high-powered laser beam that you can use to target specific subpopulations or patterns of neurons within your cranial window. So this provides us a way to perform monitor manipulation experiments in real time. So you use a single laser beam for standard laser scanning, 2-photon microscopy, and the second laser beam in combination with something like a redshifted channel rhodopsin to activate populations of cells. And there's loads of cool questions that I would love to talk to you all about that we're trying to target with this kind of approach. Okay, so what do these neurons actually do? I just want to highlight briefly that we've known for a long time now, or at least since 2014, 2015, that sensory representations can be readily modulated by lots of different things. This is one of these, choose your favorite buzzword. Some people like to use the term arousal, internal state, behavioral state, attention. All of these things can modulate the way in which auditory cortical neurons respond. And this is just a really nice example from Matt McGinley's 2015 paper. And the main point I want to make here is that as pupil diameter fluctuates, pupil diameter is a well-known measure or readout of the arousal systems of the brain. It is very, very coherent with auditory cortical membrane potential. And this has lots of effects on the way in which neurons respond to sound. And so, as with a lot of systems neuroscience labs, as I'm sure a lot of your labs, we have full-face videography attached to all of our experimental rigs, so that we can try and capture fine fluctuations in uninstructed movements, body movements, facial movements, as well as fluctuations in pupil diameter. And so just as a first pass, what we wanted to do was record the neural activity of multiple different cell types. So here we're using approaches to target layer 2-3 intratelencephalic neurons, as well as layer 5 IT, ET, and layer 6 CT cells. And we're going to just play a bunch of different pure tones to these cells while using 2-photon calcium imaging and concurrent videography. This is something that we have a preprint about this on bioRxiv, so I'm not going to go into too many of the details, other than to make the point that the effect of arousal is very, very heterogeneous. And one of the ways that we tried to show this was using an unsupervised clustering approach. So we're using hierarchical clustering here on all sound-responsive cells. So the way that we're setting this up is that you can think of this as having a three-dimensional tensor. So essentially a matrix of cells as a function of pupil. And then the third dimension there is trials. So every cell has a kind of pupillary tuning curve across trials. And what we can do is then cluster that matrix and look at what the clusters actually represent. And the main point I want to make is illustrated at the bottom here. This is the clustered evoked activity. And this is just best frequency type responses. And what we can do is actually identify what I'll call distinct functional arousal motifs. So you can find that the effect of arousal is very, very heterogeneous. You can see kind of standard inverse U type responses, which people like to talk about in the literature a lot, as well as the inverse of that, kind of what I've called quadratic on the left-hand side, as well as neurons that are monotonically increasing with pupil and monotonically decreasing with pupil. But we can then further break this analysis down by cell type and actually look at how these arousal modulation motifs segregate across cell type. And kind of consistent with the rabies result that I showed earlier, where everything projects to everything else, it turns out that although the effect of arousal is very heterogeneous on tuning properties, this heterogeneity is actually somewhat homogeneous across cell types. So we find a diversity of different motifs, you can see illustrated there, in every cell type, but just at subtly different proportions. Okay. So now moving on to the kind of behavioral side of the talk. Much of my lab is devoted to using both head-fixed and freely moving behaviors, different kinds of sensory-guided behaviors, but I'm going to focus here on just one behavior. This is a head-fixed choice behavior that I'll refer to as a perceptual categorization task. So this is a two-alternative force choice task where a head-fixed mouse is presented with one sound, and the animal has to categorize that sound as being either fast or slow. So we're using sinusoidally amplitude-modulated noise here. We begin the task with a kind of simple operant training. This is where the animal has to easily discriminate between the easiest exemplars in the stimulus set. And once the animal gets to around 85% correct, it moves on to a kind of psychometric phase of learning where we introduce all of the additional stimuli in the block, and this turns into more of a categorization task. And if we look at the psychometric functions in the bottom right, you can see that early in this psychometric phase, the slope is very, very shallow, and this becomes steeper as a function of time, indicating that the animal is actually learning this categorization. This is something that we were very interested as to whether the auditory cortex was necessary. I'm not going to go into all of the details here, other than to say that if we use a very, very potent blue light inhibitor to silence the auditory cortex constitutively, you can see that performance goes down to almost chance, and it biases the psychometric functions in a very, very specific way, which I'll talk about over the next few slides. This is not some weird artifact. If we use light on the surface of the brain and record using a laminar probe across the entire laminar profile of the auditory cortex, we can see that we're actively silencing cells throughout. So the main effect when we silence the auditory cortex seems to be through this alternating what I'll call perceptual choice bias. And this is kind of illustrated best on the top left-hand side here. Choice bias here I'm just defining as the difference between left and right choices normalized by their sum. So that's a kind of standard asymmetry index that will give you a number between -1 and 1 that indicates just how kind of biased you are to a particular lick spout. And the main thing to take away here is if we look at the light-on trials, so when we're silencing the auditory cortex, this perceptual bias gets far, far greater, and it seems to alternate from side to side as learning progresses. And we can see that really nicely in the example on the right-hand side. This is from a single mouse. Session 24, the psychometric function is very, very biased towards a particular lick spout, and then that flips just a few days later on session 30. Inhibiting the auditory cortex also affects the dependence that we have on task history. So this is kind of a nod to a lot of the really nice work that's come out of the visual psychophysics literature, that as tasks become harder, we rely more on our past judgments to guide our current judgments. So our measure here of task difficulty is essentially the distance from the category boundary. Essentially, the further you are from the category boundary, the easier the task gets because the stimuli are easier to discriminate. And indeed, if we then plot the distance from category boundary as a function of the probability from a repeated choice, you can see that in the control case, when we're closer to the category boundary and the task is really hard, we rely more on our previous judgments. If we silence the auditory cortex, this essentially just flattens out, so that even when the task is really, really easy, the animal is finding it hard. And kind of consistent with this, it seems like auditory silencing is not actually affecting sensory motor reports. This is just an example of the mouse licking on slow trials, on fast trials. But the critical point I want to make here is that if we compare light on and light off conditions, the animal is still licking. So the animal is making a decision. The animal is just making the wrong decision when auditory cortex is silenced. Okay, for the remaining 5 or so minutes, I just want to focus on what some of these particular cell type subpopulations are actually doing as a function of learning this task. So this is the experimental timeline I'm going to run you through. On the top left-hand side, this is just a schematic of the behavior. I'm going to talk about 2 different kind of behavioral contexts, an active context where the animal is actively engaged in the task, as well as a passive context, which is where we remove the lick spouts and present the exact same sounds to the exact same neurons in the exact same mouse on the exact same day. So this is kind of a well-controlled passive listening condition so that we can look at behaviorally gated effects. And the experimental timeline is on the bottom there. We're going to alternate the active and passive sessions across days, and we're going to monitor populations, large populations of these different cell types as a function of learning. So the first result I kind of want to draw your attention to is that category learning seems to reshape extratelencephalic tuning during engagement in this task. So on the left-hand side, I'm just highlighting a couple of exemplar sound responses. These are from just 2 representative ET neurons, as well as their corresponding SAM rate tuning curves on the bottom left. We're plotting them for the entire population on the right-hand side. These are just normalized matrices where each row represents a particular tuning curve. And the one thing I want to draw your attention to is the layer 5 ET active case on the bottom right. So each row here is early learning, late learning. And if we look late in learning, I hope you can see the development of these kind of two squares almost. I'll quantify this in the next slide, but you can see these two different squares in the top left and top right-hand side of the matrix there. And this is kind of implying that category learning, once the animals have acquired this task, the tuning functions are broadening and changing to represent what I'll call discrete perceptual categories. And we can kind of quantify this just a little bit by looking at the number of responsive stimuli. And again, I just want to draw your attention to this big green bar in the active late condition so that we can see that the number of responsive stimuli is peaking at 4 in the active late condition. So ET tuning is actively reshaping during task engagement as a function of learning. And so we really wanted to investigate this categorical coding. This is essentially what it looks like. We have a categorical selectivity index. This is again another asymmetry index that just looks at the difference between two responses normalized by the sum. And on the right-hand side, you can see 4 representative examples. On the left-hand side, this is a neuron that's very selective to slow stimuli and not fast stimuli, and it has a categorical index of like -0.9. And on the right-hand side, the opposite, a highly positive index corresponding to a neuron that really likes fast stimuli and not slow stimuli. And then you see everything in between. This is really the primary result that I'd love for you guys to take home with you today, because this really summarizes what is happening in these neurons. This categorical coding emerges selectively in the L5-ET population. So here what we're doing is plotting histograms of the categorical selectivity index, and we're plotting them both early and late for the active and passive conditions. And we'll start by just drawing your attention to the layer 5 ET cells in the bottom left. So although they start off bimodal, we'll talk about that in the next couple of slides, the distribution becomes really highly bimodal late in learning. So as animals are learning this categorization task, the categorical representations in these neurons are becoming more and more severe. This is something that you don't see in either of the other two cell types, but it's also an effect that is dependent on task engagement. So if we look at the passive histograms on the right-hand side, as soon as we take the lick spouts away and the animal listens to the same sounds in the same session on the same day, this bimodality just completely disappears, and you get a unimodal histogram sitting around about 0. And interestingly enough, if you then correlate the absolute value of the categorical index as a function of percent correct, it's strongly correlated in just the layer 5 ET neurons. So the categorical selectivity is changing the sensory coding properties in these neurons, and the magnitude of that effect correlates really nicely with behavior. Now, a common critique of this work has been that could this just be one big effect of motor? This is one potential confound with these kind of two alternative force choice tasks, because animals are essentially tasked with licking to report their response. And indeed, there is a potential confound between both stimulus and choice-related activity. Prior studies in the auditory system and other sensory systems have shown robust choice encoding during different tasks. But more generally, we know that there is widespread modulation by movement, by internal state of auditory cortical representations. So one way that we chose to kind of handle this in our own data set was to use a generalized linear model to separate neurons according to whether or not their activity was better explained by either stimulus-related or choice-related variables. Here on the left-hand side, this is kind of schematized quite nicely. We have a design matrix where the design matrix includes different time lag predictors for both stimulus, identity, and lick direction. And we're fitting this model to just the z-score deconvolved calcium activity. The main take-home from this, though, is that when you fit a generalized linear model like this, you end up with weights, these beta values that correspond to the different predictors in your design matrix, and we can then compute what's called a coefficient contribution, which tells us just how much one of these individual predictors contributes to the neural response. And so this gives us a way to nicely statistically dissociate both stimulus and choice. On the right-hand side, at the top there, we have what we're calling a stimulus neuron. This is a neuron with a high coefficient contribution for stimulus and not choice. And in the bottom right-hand side, we have a choice neuron where the opposite is true. And what's really, really cool is that we can then ask whether or not the learning-related increases we're seeing in categorical selectivity actually does reflect genuine changes in sensory encoding, or is it just a byproduct of choice-related activity? And after we classify neurons, again, on this kind of per-session basis as either stimulus or choice, we can recompute the categorical selectivity index and replot these histograms. And if we restrict our analysis to just the stimulus neurons, you can see that this, you end up with a very nicely unimodal distribution early in learning that then becomes bimodal late in learning, only in the stimulus neurons. So this seems to be a genuine change in sensory encoding within these neurons, and it doesn't seem to be a byproduct of choice-related activity. However, it turns out that these neurons encode both category and choice. I'm not going to go too much into the details here, other than to say that we can use machine learning decoding strategies to actually ask how well these neurons can predict category and choice. And these neurons essentially multiplex this information. They can encode both the identity of the category that was presented as well as the choice that the animal made. And I'll just finalize with one little neat analysis using the boundary stimuli in our data set. So this is one little wrinkle that I haven't actually talked about yet. We introduced a boundary stimulus right at the category boundary. So this is a stimulus that is fundamentally uncategorizable. It's neither fast nor slow, but we reward it probabilistically. So we ask the animal to still make a decision based on the identity of this stimulus. And essentially, the result here is on the right-hand side. If we use a little bit of dimensionality reduction, here I'm using a PCA plot. If we take a population of expert ET neurons, reduce its dimensionality down to 3 dimensions, and plot the responses to the exact same stimulus dependent on what the animal thought the neuron was, the trajectories go in vastly different directions. And we're quantifying this on the right-hand side using a Euclidean distance, and you can see that this changes as a function of learning, and it's far greater in ET neurons than any other cell type. Okay, so I'm almost out of time, so I'll just finish by acknowledging my lab. Megan Arnold did a lot of the anatomy work, Keith Kaufman did a lot of the pupil work, Becca Crowell did a lot of the behavior work, and I just want to give a special shout out to Nathan Schneider, who's somewhere in the audience. Nathan is a superstar graduate student who did a lot of the in vivo recordings, all of the cell type specific behavior stuff at the end. He is maybe going into the job market soon, looking for postdocs. He's kind of in his twilight years in my lab. So please go and talk to him. We also have a poster focused on all of this behavior stuff, which is why I was kind of lacking on some of the details. So go harass Nathan. I think it's on Tuesday, and he'll present a whole bunch of additional results that I didn't have time to talk about. And then I'll acknowledge my funding. Thank you. Can I throw it?
So I received a request that if you don't want this to be thrown at you, we will come and hand it to you. So don't worry.
I was really waiting for Jonathan's hand to go up because I wanted to throw Oh, no, okay, sure. Yes.
And maybe we can invite questions from trainees first. So if you are a trainee and you have a question, don't be shy, raise your hand. Introduce yourselves, whether you're a trainee or not, and ask a question.
I don't know if I can, Kishore has a question, but I don't know if I can launch it that far.
Hi, Ross. Thank you very much. It was a beautiful talk.
Thank you.
What do you think is a teaching signal for these IT neurons?
Yeah, the honest answer to that is that's one of those questions that I can give you an answer in a few years. The next stage of this project is trying to identify a top-down circuit mechanism that is actually guiding these changes. What I can tell you so far is that, so this is a project that another graduate student in the lab has taken on. We're essentially tackling it as a first pass, because I'll step back and say that every lab, including many in here, always ends up with their kind of favorite region, anterior cingulate, orbital frontal, blah, blah, blah. We're kind of taking a more kind of general approach and using a kind of Svoboda-esque tactic. We're clearing the skull, guiding an LED to different points in the brain, and then looking at how different top-down projections can differentially affect both passive listening and active listening. What I can tell you so far is that because everything's connected, it's perhaps unsurprising that if you tickle any one of these areas, you can drive changes in tuning functions. The biggest effects that we've seen so far seem to be associated with both anterior cingulate and orbital frontal. But those results are very, very preliminary, and that's only in passive listening. We've not got to the point of doing these experiments with behavior. So yeah, top-down circuits to be identified.
Hey, Ross.
Oh,
great talk. So one question that came up for me when listening to the whole thing was, in the first half, it seems as though these 3 excitatory cell types have sort of input promiscuity, in the sense it's pretty similar.
Yeah.
They have a similar sort of change in activity based on arousal. But then in the second half, they have this very different response in a categorization task. So could you speculate on what you think is driving that? Is there a different, how can you have both in the same sort of group?
I think ultimately it's going to come down to, I mean, again, the answer is ask me in a few years because we're trying to work this out. I think it just comes down to behavioral gating, right, and differences between active and passive engagement. Because, like as I just mentioned there, a lot of the passive listening experiments we've been doing when we tickle these different regions in the front of the brain, it all modulates the auditory cortex, and you can change tuning functions. But that doesn't tell you anything about what actually happens when the animal is engaged in a task. So,
but I guess more specifically, your arousal study suggests, well, IT, ET, CT neurons all are modulated the same way, right? Engagement that you just mentioned, one aspect of that is arousal, but in a way you can rule that out now, right? Because that can explain the difference between ET and, let's say, CT neurons or IT.
Yeah. I mean, that's a hard one to answer because pupil diameter is really messy. It's a core signal that correlates with absolutely everything that people have looked at. It correlates with every neuromodulatory system that people have looked at. The way in which it correlates is slightly different. So I think it's kind of really hard. It's a good thought experiment, and it's something that we've tried to think about. But yeah, I don't have a good answer for you, to be honest.
Hi. Thanks a lot. That was nice.
Thanks.
A couple of questions. So for the categorical projections you find at the very end of the talk,
say that once more.
So, for the categorical projections, the categorical responses you found at the very end of the talk, is it lateralized in the sense that the left sound, you find responses to that sound more often in the right hemisphere?
Oh, no, we flip it.
Okay, cool.
Yeah.
And my second question is for the bias you obtain with the opto inactivation. So is it something that is evolving slowly across days?
Yes.
Okay, so there is like some history and...
Yeah, so, well, there's two answers to that. First off, we didn't do opto while the animals were actually, while we were recording from these cell types. So we can't make that particular link yet. That result was a behavioral study with optogenetics. There is a lot of stuff, actually, this is a question that I can direct to Nathan because there is a lot of choice history dependence that we looked at that I didn't have time to talk about. It's an entire figure in the preprint and it's going to be on the poster tomorrow. So if you talk to Nathan, there's a lot of information there about that.
Thanks.
I'm trying to relate this to kind of how I think about things. So, we've done a lot of work, and a lot of people have done a lot of work looking at how responses change across the cortical hierarchy, so much coarser scale, and you tend to find that as you move up the hierarchy, responses become more selective for high-level things like sound categories, like music and speech. Those effects are, they're presumably tied to behavior at a high level, but they seem to be pretty stimulus-driven, right?
Okay.
So it's not like only when you are responding, pressing a button when you hear a particular category, you see these responses. And so I'm trying to relate this to what you're kind of talking about, which is at a much finer scale. It seems much more tied to behavior. I sort of don't quite know how to think about the computation that's being performed. You know what I mean? Like, how does that relate to how we think about how these neurons compute more complicated things? So I'm sort of asking, how do you relate those two very different worlds or think about them? So.
Yeah, I think about this in terms of sensation to action, right? At the end of the day, if you get an animal engaged in this kind of behavior, you're converting sensation into a motor action. And to me, that's very corticostriatal in nature. And this is something that, again, I didn't really focus on too much. But there's a lot of really interesting differences between these different cell types and which parts of the basal ganglia they talk to. The short answer is that it turns out that these ET cells project primarily to the tail of the striatum, which is the part of the striatum that is more involved, less habit formation and early learning, and more the kind of refinement of stimulus response contingencies. So I think it's very, very cortical striatal in nature. As to the level of the computation or what kind of computation is actually occurring, we don't really know yet. We do see a lot of, in separate studies, we've actually looked at how stimulus coding changes across the hierarchy. Actually, I think we have a poster on it at SFN if you're going to be there. Noise invariant representations, for example, change in a very consistent way as you move through this canonical cortical hierarchy from layer 4 to 2, 3, layer 5, where a lot of these kind of noise invariant representations seem to become prevalent at the output layers of the cortex. So we do see stuff that's changing, but yeah, the vast majority of the behavioral effects we see are located in layer 5.
All right, I think we'll end it there. Thank you very much.